Hi, everyone. I'm going to talk today about a project that I did in 2018 in Acadia National Park looking at cross-ecosystem subsidies and how invertebrates can help us understand mercury exposure in songbirds. So what I've been interested in for a while is this idea of cross-ecosystem subsidies, specifically an aquatic to terrestrial subsidy. So what you can think of this as is insects that start their life cycle in the aquatic system and then at some point emerge to become winged flying insects. And once they're out, they can be consumed by riparian songbirds. So this kind of flow of nutrients is really important. It provides extra biomass of invertebrates for riparian birds, which can be a huge positive and concentrate a lot of birds in that riparian area. Where it becomes a problem is in my research where aquatic water bodies can be contaminated with mercury. And so with this, mercury gets into the bodies of those emergent aquatic insects and then moves out into the terrestrial ecosystem. So here, now we can see that these subsidies, which are really great in a lot of ways, can also move aquatic contaminants into the riparian zone. And we see that riparian songbirds are often exposed at high levels. So I spent a lot of time thinking and working on what influences songbird mercury exposure. At the start is just, is there mercury in the system and is it methylated? This is kind of the process by which mercury gains a carbon group, and that's how it becomes bioavailable to organisms. So that's kind of the first phase, is just, is there mercury in the system? Also, the foraging preferences of the birds are really important. I've been studying not aerial insectivores for the most part. I study things like song sparrows, warblers, things that can have more flexible foraging preferences. So looking at what they're eating can influence their mercury exposure. And then also just the actual flux of these emergent aquatic insects into the riparian area. How many of them are actually getting out of that water body to be potentially consumed by songbirds? That is influenced by the relative health of the aquatic system, the difference in aquatic habitat, and even things like the presence of fish or other predators that might eat these emergent insects before they emerge to become food for songbirds. So what we kind of have is a system where if we look at it roughly, we have mercury bioaccumulation on one axis and insect emergence on the other. So in areas where you have high mercury bioaccumulation and high insect emergence, there's very high risk to songbirds. In areas where there's low mercury bioaccumulation and low risk and low insect emergence, we have low risk to songbirds. But then there's also kind of these other quadrants where, what if you have high insect emergence but really low mercury exposure? Could birds still be at risk just by the magnitude of insects that are getting at? And what if you have high mercury exposure but relatively low emergence? Is this still a risk to the birds? And in general, the more I work on this, the more I realize that aquatic subsidies have this kind of real PR issue, I like to call it, where it's kind of this very important nutrient pathway. We want to have healthy ecosystems that produce emergent aquatic insects. It's a great nutritional food source. Often these are pulsed in temperate regions early in the season when terrestrial prey, such as caterpillars and other bugs, may not be as abundant. So birds are using this resource early in the season before leaf out. But we don't want to be exporting aquatic contaminants. And this is kind of how they're getting that free ride out of the aquatic system into the riparian food web. And then kind of the other wrinkle where I say this is a PR issue is that a lot of people don't particularly like insects and spiders, especially those such as emergent insects that seem to be synonymous with mosquitoes or black flies, especially if you're living in Maine. So people don't necessarily have this positive connotation of the bugs that are coming out of the water, even though they're not all mosquitoes, and a lot of them are these very important food source for a lot of riparian animals. So I studied this kind of problem in Acadia National Park. Acadia is a great place to do research because they're, for me in particular, very concerned about mercury pollution, but also they're kind of interested in that like PR issue that I talked about. So they really want to see how to get visitors engaged with the resources to kind of understand the type of research that's going on in the national park, and also just take part in science education to get people more literate about the type of research that we do. So I was funded through the Second Century Stewardship Fellowship, which was brought together to bring early career researchers into the park to do this type of science. I'm very thankful for it. It's been a great opportunity to kind of learn and do some things that are a little bit different for me as a researcher, as you'll see in this talk. So I picked 4 study sites in Acadia. These were chosen in a lot of ways for ease of access because we were going to bring volunteers into these different sites, but they were also kind of representative of different habitat types. So Gilmore Marsh is kind of this broad, open marsh complex. Marshall Brook is a small stream with wetland, which we'll get into more. It's fairly degraded, having a lot of kind of urbanization around it. The Schoodic Beaver Pond is a small beaver pond complex that we kind of focused on one of them on the peninsula side of the park. And Hunter Brook is a kind of fast flowing mountain stream that dumps directly out into the ocean. So part of the reason I focused on these sites was that researchers had already been studying them for many years through the Dragonfly Mercury project. So this is a project that is getting people, getting community scientists out into parks to collect dragonflies and send them back in for mercury analysis. This is nationwide across the US, trying to look at trends in mercury exposure. So I pulled out these 4 sites to kind of show you the data that I was working with previously. So this is dragonfly mercury from the 4 sites that I chose. We can kind of see the 4 sites kind of ran the spectrum of sites in Acadia. Schoodic had some of the highest dragonfly numbers in Acadia, and Gilmore had some of the lowest. So we see kind of that trend. So what I wanted to do was kind of collect invertebrates both in the aquatic and the terrestrial ecosystem using 2 different methods. So the first method was using researchers. So my researchers in this case were me and a bunch of college students from Purchase that came out with me. And we did kind of quantitative, more traditional invertebrate sampling. So our terrestrial sampling was a variation on like a branch analysis or quadrats. We did 10 standardized samples per site per week using beat sheets and sweep nets. The aquatic sampling was dip, net, quadrants, again, 10 standardized samples per site per week in each of these. But then the other side of it, which was rather unknown to me, was that I wanted to also use volunteers. So these are park visitors who were going to come out and help us collect bugs. So we constrained them to the same areas as we did our researcher collected samples, but we didn't have them work in quadrats or anything like that. That's a rather tedious thing. And we wanted to be able to basically show everyone the cool part of science, right? Like once you get into science a little bit more, you get to see the tediousness, but we literally just brought people out to enjoy the park, to collect bugs, get to see them, get to sift through mud. We even, the way it worked, we didn't have them come into the lab with us. So like my students were the ones who actually did the sorting and the counting later. So a lot of the actual volunteer effort was the fun, getting into waders, experiencing the park in this different way and seeing the bugs that we sampled. So we had 2 different research questions. Kind of on the scientific side, I wanted to see, does the mercury in birds follow the same trend among sites as mercury in dragonflies that had been collected from the Dragonfly Mercury project? Does the biomass of aquatic and terrestrial invertebrates vary among sites? And does the abundance of emergent aquatic insects vary among sites? So in general, does biomass difference, does the actual number of emergent insects differ? And does this magnitude of emergent aquatic insect flux influence mercury exposure in those riparian songbirds? And then kind of on the visitor volunteer engagement part, I wanted to see, can we recruit park visitors to help us collect bugs? Is this something that people would be interested in? And will volunteer collected data be similar to researcher data? So the first question, does mercury in birds follow the same trend among sites as mercury in dragonflies? So remember, this is the Dragonfly Mercury project data, and then I also went out and sampled birds at each of these sites. And in general, we don't really see this great correlation in terms of high sites and dragonflies being high sites in songbirds. For the most part, sites with the lowest dragonfly mercury have the highest bird mercury and kind of vice versa. Again, I'm not doing any sort of, this is literally just looking at it this way because there's a lot of flaws in comparing these data. The dragonfly data was collected in totally different years, slightly different areas, and you don't want to like compare them perfectly. But in general, we don't see this perfect correlation between dragonflies and songbirds. In particular, I want you to kind of think more about this Marshall site here, which had relatively high dragonfly mercury, and it seems to have very low songbird mercury. And so I'm going to kind of get into what could be going on there. So can we recruit park visitors to help us collect bugs? Yes, we had a great turnout. I recruited on social media, through the park website, through just different contacts at schools. And we got great turnout for all of our kind of volunteer collection days. So we did 3 of these at each site throughout the summer and got a whole bunch of people. Our low end cutoff for the children was 7 years old, and we got up, up until retirees. And I think our very easy data collection methods really help make family participation possible. I had to communicate a lot with people beforehand via email, and a lot of them had questions of, will my kids want to do this? Like, how hard is it? And we kind of could just say, your kid can do as much or as little as they want. And so a lot of people came out with family members who said that they didn't want to ever touch a bug, and in the end did touch bugs and have fun. So I think our kind of loose data collection for our volunteers allowed us to kind of have this experience of bringing entire family groups out into doing the data collection. So looking at our researcher data, I wanted to see, does the biomass of aquatic and terrestrial invertebrates vary among sites? And the answer is yes. So on the top here is the aquatic sampling, on the bottom is the terrestrial, and we see variation among the sites, which likely I'll be able to tie to habitat differences between the sites. But will volunteer data be similar to researcher data? Remember, it was very open in how they collected data. And when it comes to biomass, we don't see necessarily the same exact trend when we're looking at the volunteer data. I think a lot of this has to do with just volunteer effort and the fact that we didn't constrain them to quadrats. And again, like this effort was also because we had different numbers of people out each day. We had variable numbers of volunteers each day, and the volunteer range in their attention to detail. So we kind of see that could potentially, I tried to look at, did the just number of participants influence the biomass of terrestrial invertebrates? Not in any sort of strong, perfect correlation. I think this is because not only is it the number of participants, it's how hard the participants actually wanted to work. And I had a great time doing this. I fully support the idea that we got people interested in bugs and we didn't make them hate science more. So I'm totally okay with the fact that we didn't standardize perfectly. So does the abundance of emergent aquatic insects vary among sites? And yes, this is where we're going to start to see large differences. So this is, again, our researcher sample looking at aquatic and terrestrial sampling, but I've parsed out the difference between a fully aquatic and a emergent aquatic bug, and a fully terrestrial and an emergent terrestrial bug. So you can see here, kind of that Marshall site stands out as potentially this difference where they have both in the aquatic sampling very low numbers of emergent aquatic, and in the terrestrial sampling very low numbers of emergent insects. So again, will the volunteer data be similar to researcher data? What I did is, because we know we have that biomass issue, I just looked at relative percentages between these for the volunteer samples, and we can see some variation. I have a hard time knowing if this is something that we would want to use as scientific data, or if it's flawed enough that it kind of is just, I'm going to use it in a different way, which we're going to get into. So what could be going on here? We know from the Dragonfly Mercury Project that dragonflies are great indicators of mercury in the aquatic system. They correlate really well with fish mercury, which is something that's really important for human health, so that you can sample a dragonfly and potentially know if fish are going to be contaminated. And we know that songbirds indicate mercury exposure in the terrestrial system, but why don't we see this better correlation between them? I think the answer is that mercury is mitigated by a filter of emergent aquatic insect flux. So in particular, that Marshall site, which had relatively high mercury in the aquatic system, didn't have very much flux of emergent aquatic insects out into the terrestrial. And so therefore the birds had relatively low mercury exposure. So when I look at this kind of broad strokes again, does the magnitude of emergent aquatic insect flux influence mercury exposure in riparian songbirds? We start to see some sort of trend where the average number of emergent insects per sample seems to be correlated with the average bird mercury, mostly driven by that Marshall site again. So hard to say without more data what all of this means, especially when you look at the amount of error associated with each of those, where we see these really variable mercury numbers, especially at a site like Schoodic, where it's variable both in emergent insects and bird mercury. So there's just a lot more that we need to do to kind of understand this system. So future directions, I talked about how I'm not really sure if the volunteer effort gives us great biomass or abundance counts, but we did collect a lot of insects. And so what I realized we could do is kind of use those samples for running analyses, because we have a large kind of amount of insects. So I've already run them for stable isotopes, and I'm going to run them for mercury, because this will give us a better idea of how the food webs are kind of set up. And if you look kind of at this, this is really preliminary data, but we can see potentially differences in how the food webs are kind of structured between the 4 sites. Especially something is going on interesting in Marshall, where the aquatic samples are very enriched in nitrogen. So this is something that I can dig into more, and it's exciting to me to be able to use all of that effort that the volunteers put in and get some really cool results from it. So I'd like to thank all of the undergrads who worked on this project. It was a hard summer in 2018. I broke my ankle halfway through the summer, and a lot of these students really stepped up to kind of lead a lot of the volunteer efforts and collect a lot of the bugs. So I'd like to, call them all out and thank them all, many of which are graduated from the program now. And then also acknowledge my funders. So again, that Second Century Stewardship Fellowship is an amazing opportunity for me to be able to do this work. And then my college also kicked in a good amount of money to kind of support me and the students. If you want more info, our website is acadiabugproject.com, and you can contact me. I know this was kind of rushed, and I'm really sorry if you could hear my dog chewing on his bone through this whole presentation. I decided to just keep going, even if it was happening. And I'm happy to take any questions live.